Morning. Today we are going to start chapter number 5, part 3, Scene Ecology. So, see here that it is the study of ecology at the level of community or ecosystems. It can be defined as the study of interactions between species in communities on many spatial and temporal scales, including the distribution, structure, abundance, demography, and interactions between existing populations. It aims to investigate the relationship between different species that form a community and their interactions with the surrounding environment. Scene ecology involves both biotic and abiotic factors. The abiotic factors, that is annual temperatures, soil pH, can influence the way species interaction with each other. Abiotic factors filter the species that are present in the community and therefore community structures. For example, the difference in plant presence in the desert compared to the tropical rainforest is controlled by the annual precipitations. These non-living factors also influence the way species interacts with each other. Human can also have an effect on community structure through habitat disturbances such as introduction of enhanced species. Scene ecology helps us learn about our surroundings and how various organisms interact with each other, whether peaceably, territorial, or aggressively. Scene ecology has its roots in European plant sociology. It stems from phytosociology, which is the study of plant communities. These communities were categorized with specific systems, allowing for easy access to the information. Scene ecology does not, but on a much larger scale, focusing on all communities in nature. Modern scene ecology focuses on patterns within the communities of species and organisms. This includes how many of each species, how much stake each species has in the community, what the species does for the community, as well as how the species eats within the community. It also can get into predatory prey dynamics and how it affects everything listed before. There are a few theories to go along with scene ecology. Holistic theory, created by Frederick Clement, is when species in a certain area, though near each other, are mostly independent from each other. Individualistic theory was created by Henry A. Gleason. He created the concepts of an open community. When most populations changes according to the environment, these changes happens to individual populations and not all populations equally. This creates communities that would be labeled as a discrete or almost provides communities. Natural theory, made popular by Stephen Habel, is a hypothesis that does its best to explain abundance of certain species in ecological communities. His hypothesis makes assumptions such as that members of an ecological community have differences through none of them are relevant to their success. This means that biodiversity would occur on a random scale, which causes much controversy in the scientific community. Scene ecology is the basis for how we view communities. Communities exist all around us, from our plants and insects to the microscopic organisms we cannot see with our eyes. Scene ecology helps better understand these communities, and with that will help us adapt our lives around it. Scene ecology is subdivided into two principal branches: aquatic ecology and terrestrial ecology. There are subdivided further into many branches. These are illustrated in following figures. Then next point, that is species diversity, definition, concepts, scope and types, alpha, beta and gamma diversity. So first we see introduction. Species diversity and biodiversity are widely used terms in ecology and natural resources management. The term biodiversity is widely used but rarely defined well. Different authors have defined it in their own way and preparation. It is most commonly defined as the number of species of a particular geographical area. Is it simply the number of species, or is it something more? Our planet Earth is full of different kinds of organisms. Each of these organisms are unique with differences and variations among them. These differences and variations are referred to as diversity. The diversity is the vital characteristics of all living organisms in their multiple levels of organizations. The most fundamental differences among the organisms are generally recognized in their levels of biochemical and genetic constituents, morphology and ecological role. Such diversity of living organisms constitutes all levels of natural variations, such as from genetic materials, that is genetic diversity, from roles, functional diversity, to ecological processes including their interactions. The synthetic approach of all these variations is called biological diversity. A conservation biologist, Thomas Loja, used the term biological diversity during 1980s. Walter Rosen has coined the term biodiversity in 1986. Since then, the term biodiversity is of widespread use among biologists, environmentalists, political leaders, and all concerned persons worldwide. Then we must see definition. Species diversity refers to the variety and number of different species that are represented within a community or ecosystem at a given time. Simplest measure of species diversity is a count of the number of species present in an area. That is, species richness. Ecologically, species variety is measured by species richness, species composition, that is a list of species or species assemblage, and relative abundances of species. In other way, it can be defined as the number of different species present in an ecosystem and relative abundance of each of those species. Diversity is greatest when all the species present are equally abundant in the area. Species richness and evenness also are two components of species diversity. If the number of individuals within a species is fairly constant across communities, it is said to have a high evenness, and if the number of individuals varies from species to species, it is said to have low evenness. High evenness leads to greater specific diversity. Species diversity is a function of species richness. The number of species in a given locality and species evenness, and the degree to which the relative abundances of species are similar. Tropical areas have greater species richness as the environment is conducive for a large number of species. The next point, that is concept of species diversity. Most of the plants and animals exhibit variations consumed by the patterns and processes of species formation. Within a population, variations can be continuous, discontinuous, or genetic. Variations are also observed in the time and space between successive generations, seasons, and also among allotropic populations. Species generally exist a natural entity possessing certain common characters which are distinct from their close relatives. Therefore, there is a spatial and temporal variations in species distribution. The concepts of species diversity span a range of organizational level. It means that it is important to specify how it is being used. Species diversity is important characteristics of community structure. Species diversity is an appropriate terms for ecologists who are interested in understanding the mechanisms and effects of certain ecological phenomena, such as population, environmental disturbances, etc. Diversity within species is genetic diversity. Diversity between species is species diversity, and diversity at ecological or habitat level is ecosystem diversity. Some ecologists have included a fourth form of diversity called landscape diversity. Early naturalists were quickly observed that tropical areas contained more species of plants and animals than did temperate areas. To describe and compare different communities, ecologists broke the idea of species diversity down into three components, that is alpha, beta, and gamma diversity. Okay, then we must see the scope. Species diversity is an important tool in biodiversity studies and widely used for conservation. It helps to assess the homogeneity of an environmental as well as the determine of sensitivity of the species and ecosystems. Biological diversity is essential for human survival and economic well-being and plays a important role in modulating ecosystems, functions, and stability. Each species has a role in the ecosystems. Various species provide us not only with food but also contributes to clean water, breathable air, fertile soil, climate stability, pollution absorption, building materials for our homes, prevention of disease outbreaks, medical resources, and more. Species diversity also contributes to ecosystem health. Okay, then after that we must see next point, that is types of species diversity, that is alpha diversity, beta diversity, and gamma diversity. So first we see what is meant by alpha diversity. Alpha diversity refers to the average species diversity in a habitat, or it is just the diversity of each site. It is a local measurement, the number of species found in a particular area or ecosystems. Alpha diversity describes the species diversity within a community at a small scale or a local scale. Generally, the size of one ecosystem, for example, habitat A has 9 species of herbs, therefore the alpha diversity for herbs at this place is 9. Alpha diversity is quantified by indices and by ranks abundance models. Then second type, that is beta diversity. Beta diversity refers to the differences in the species composition among two or more sites habitats. It refers to the ratio between local or alpha diversity and regional diversity. It is the variations of species composition between two habitat or regions. It considers that the alpha diversity of the habitat and the number of unique species on each habitat. It is at a larger scale and looks to compare the species diversity between two separate entries that are often divided by a clear geographical barriers like a river or a mountain ranges. It is also defined as an index of the rate of increase of alpha diversity as new habitats are sampled. So is a measure of turnover of species along spatial gradients. Beta diversity links alpha and gamma diversity. It is calculated by the following equations. Beta diversity is equal to number of species in habitat 1 minus number of species common in habitat 2 and 1 plus number of species in habitat 2 minus number of species common in habitat 1 and 2. Then third type, that is gamma diversity. Gamma diversity is the measure of all of the overall number of species of the entire landscape, that is regional species pool. It is the total diversity of a landscape and is a combination of both alpha and beta diversity. Gamma diversity is the full diversity of an entire sample landscape or gradients. It is basically the sum of all the species of all habitats within the region of interest. Gamma diversity is studied at a very large scale, say a biome where species diversity is compared between many ecosystems. It could range over areas like the entire slope of a mountain or the entire littoral zone of a seashore. Hunter in 2002 defines gamma diversity as a geographic scale species diversity. Okay, thank you.